who will tell us both theory and practice of doing common variant associations. We're running a little bit behind, so we're maybe 10 minutes into the break. I hope that's okay with everyone. So thank you, Derek.
Thanks, Jason. So I'll try to be a little bit quicker too. Is it the Mac or PC? Oh, there we go. Right, so as Jason mentioned, I'm going to be giving a more practical guide to how you can analyze and process your data and also some of the common pitfalls that you might encounter, some of the things that you might need to check in your own data sets. That way you can take some of this back to the lab and apply it to your own data. For the purposes of this talk, though, we're going to be using the ADNI genetic test data set, and so actually a lot of the commands that I'm giving in the slides can be used directly for processing those. So you can practice on the ADNI data set on your own, and also I think you can extrapolate those to your own data. Some of the things that we're going to be talking about are quality control procedures for raw data, so applying tests of missingness, tests of relatedness, filtering for things like minor allele frequency and deviations from Hardy-Weinberg equilibrium, and then also talking about the importance of looking at ancestry in your data set. I'm not sure that we'll eventually have enough time to go over this, but we might. But I also have some code that's freely available on GitHub that you can use to do image-wide genetic analysis. So I think that this has been covered pretty well so far, but just a reminder of what we're doing here. We're talking about genetic associations with SNPs. So common variants are SNPs, which are single base pairs in the genome. And as an example, just a toy example here, we're interested in brain size, so the little guys with the blue heads. And then we've collected a pretty big sample and then also collected DNA. And we're scanning along their genome and finding a location where people vary. And then taking the brain size and plotting it against which genotype they have. And so this is kind of just a really simple example, but it's a clear linear relationship. Each additional A allele gives you an increase in brain volume. So this is just a quick reminder of some of the software that I'll be talking about and that you'll need. This is also the path to the GitHub account that has a lot of the scripts that I'll be talking about. So I think that all of these are already available on the OHBM website, and then also they'll be available on Tom Nichols' website after the meeting. So you'll have all of this easily accessible. Kind of a delay, sorry. Okay, so just really quickly then, the ADNI dataset is a really great imaging genetics dataset. It's the Alzheimer's Disease Neuroimaging Initiative. I think this is a really great dataset for getting your feet wet in imaging genetics. It's freely available. You do have to sign up and submit a proposal. It's free-ish. So you do have to sign up and submit a proposal, but it's simple to do. And once you're there, there's a image data archive, the IDA, Loney IDA, and from there is where you can download all of the data that's available through the study. Here's actually just a Plink formatted file, which I'll go over in a minute. This is all the genetic data that's available for the first version, the first phase of the ADNI study. So let's get started in thinking about some of the things that we'll need for doing our quality control testing and analysis. One of the nice things about the ADNI data, is that it has quite a few problems, the one that you directly download from the website. So it's a good example for us to go over, because I think there in a lot of ways there's a lot of problems that we should discuss. One of them is that when you first download the Plink file, it doesn't have any information about diagnosis, and that'll actually come in handy knowing the diagnosis when we do Hardy-Weinberg equilibrium testing, and I'll go over exactly why that is in a minute. I provided at the GitHub link that I mentioned before a file that tells you the patient and control status of each of the subjects that are in this Plink file that we're talking about. I won't go too detailed into the Plink commands, but basically everything that we're doing here for manipulating files and filtering and things like that are all going to be done in Plink. It's your best friend. It's a really great tool for GWAS analysis. This is an overview of what the files look like right now. 750 subjects. We're starting with 449 males, 308 females, and then this is the breakdown by diagnosis. MCI is mild cognitive impairment, AD is Alzheimer's disease, and CTLs are controls, healthy controls. Right. So this is actually each of those files that I just mentioned back here. Sorry, this. Having a little bit of trouble. Okay. Each of these files are so-called Plink formatted files. Plink is a really well-documented program, and so you can find a lot of information about them online. And I have a link at the end of the protocol, but at the end of the slides. But I kind of wanted to go over what the files look like. This is a basic breakdown of what the bed file will look like. The first column is the family ID. The second column is the individual subject ID. The third is the mother's ID, father's ID, and the fourth, the sex, the phenotype, and then allele 1 and allele 2. This will obviously be different by study and things like that. And here, actually, the ADNI study is a population study. So this is sort of a pedigree file. If you had some family relatedness, the formatting of the file would look different. People that were related would have the same family ID, and they would also have coded a mother and father ID. But here, because they're all related, are all unrelated, each person has their own family ID and also their own subject ID. This is what the BIM file looks like. It's a map file. The first column is actually the chromosome number. The second is the RSIDs. This is a common way of referring to SNPs, is by their RSID. And then the second is a sort of disused column of zeros. The second is the distance or position in base pairs. And the fifth and sixth are allele 1 and allele 2. And then the family file. So this is similar to the first few columns of the pedigree column. And this is after we've now updated the file with diagnosis. The diagnosis column is the sixth column, and the sex is now included as well. This is kind of like the standard format that you'll see in a lot of genetic programs, even beyond Plink, is this setup that goes family ID, individual ID, mother ID, father ID, sex, and then a phenotype. So that's a pretty standard format, so just thought it would be important to go over. Okay, so now that we've sort of set up our file and we have everything, we need to start checking for problems. The first one to do is actually to check for discordant sex information. So I think most people are aware that men have one copy of the X allele or X chromosome and then a Y, and women have two copies of the X chromosome. And how that plays out in when you're genotyping SNPs in that area is that men have to be monogenic at that point, or have a single have a single genetic variant, whereas women have 2 copies. And we can compare the genetic sex based on the percent of heterozygosity at each SNP versus the reported sex. So this is kind of interesting. In ADNI, we run this analysis on this data set, and so this is how you do it. We actually found that there were examples of 2 subjects that had miscoded sex information based on comparing their genetic sex to their reported sex. So mistakes like this, especially in bigger data sets, do happen, and it's really important to do this check, even though it seems like something that might not be something that you would make a mistake on. I think in this case, with the ADNI data set, we weren't able to go back and resolve this issue, so we added them to what's called our remove.txt, and I think throughout the next couple of steps, you'll see we'll be adding some additional subjects based on the criteria that we're going through. Oops. Next is the test of missingness. If you remember from Sven's talk, there was talking about the different bead array chips and how, based on their color, you can determine if they're homozygous or heterozygous. In some cases, it's actually difficult to determine what a specific genotype should be. And in those cases, they're given a score. In each case, they're given a score about the confidence, how confident you are, or the algorithm is about calling a genotype for a given person. And we tend to filter, at least for Illumina chips, at a genomic call rate of less than 0.15. So we get rid of ones that we're not very confident in. A result of this, though, is that we have blank spots for subjects across the genome. But one aspect of this, actually, though, is that if there's too much missingness, it could indicate a problem in the quality of the genotyping. So one sort of unspoken threshold is actually if there's greater than 10% missingness, then the subject should be removed. And in the ADNI, we actually did find one of the subjects that had greater than 10% missingness. If you can see here, this is the column that describes missingness. It was just slightly above 10%, but to be on the safe side, and because it's such an outlier, we decided to that is probably best to remove it. So we add them to our remove.txt list. Another thing that we want to do is actually to identify related subjects. This is a population study, so a lot of the statistical models that we're using assume that your subjects are unrelated, that they're statistically independent from each other. So relatedness can actually cause a lot of bias. One of the things that we can do then is to use the genotypes to calculate how similar all subjects are to all other subjects in your analysis. And you do that using an IBS analysis. First, actually, the best way to do this is actually to prune down high LD regions. From Sven's talk, he did a really great job of describing LD. I think I have another reminder. It's slightly blurry, sorry, but you end up with these large blocks in the genome that are very similar across subjects because there's hotspots, and they tend to leave large blocks that are all passed down together. A result of this is that the SNPs within those blocks are very highly correlated. And because of that, if you have a large SNP density, if there's several SNPs within an LD block compared to, say, this one, which only has a few, and you're doing this IBS analysis, it can bias it. So one of the things that we do is actually to just select a single representative SNP from each LD block and use that to calculate relatedness. Okay, and then these are the commands. Like I said, I won't go over them in too detail. You'll have these in the PDF afterwards. But to see the results, one also general but unspoken rule is that we're filtering on IBS scores with greater than 0.2. An IBS score is basically a relatedness, a kinship coefficient. And so monozygotic twins, for example, are 100% related. So you would expect them to have an IBS score of 1. Siblings share about 50% of their DNA on average, so you would expect siblings would have a score of about 0.5, and so on for different familial relationships. In the case of ADNI, we actually found that there were 6 subjects that were related. This is actually a big problem, and so this is the column. The pi hat score is the score that we're interested in, and it looks like there's about 3 sibling pairs, and we need to handle that. So what do you do actually if there's sibling pairs? You don't have to throw out all of the data actually. You can pick one of them from each pair, so that the ones left in the analysis are still all unrelated. How you choose is up to you. I sometimes will look to see which has better genotyping based on the missingness rate, and choose the one with the best genotyping rate. Other than that, it's really kind of a random choice. So we're adding those to our remove list. And actually, in Plink, now that we have these sort of major structural things taken care of, we just apply a remove command and end up with a new version of our data set, those .bed, .bim, and .fam files that we looked at before with those subjects removed. Now we can actually start to apply some filtering. There's 3 really important steps, which are .maf, --geno, --hwe, which I describe on the next slide a little bit more. --maf is actually minor allele frequency filtering. This can remove rarer variants. Depending on your sample size, having rare variants when you test an association can actually cause some problems because outliers can possibly drive your association. So you might end up seeing a single subject with Alzheimer's disease versus controls that has some genotype and have an astronomically low p-value. So to avoid that case, you can filter out based on the frequency of how often the minor allele appears. One guideline is about 0.01, which is a frequency of 1%. If you have a smaller sample size, you would need to raise this up more to maybe 0.05 or 0.1. Similar to the idea that we were talking about before, where there's missingness across a subject based on the quality of the genotyping, there's also the possibility of having missingness across a SNP. So if there's, which could indicate a number of problems, possibly if you're using a chip that's not really meant for the ancestry that you're studying or something like that. You can use this --geno command to see if there's greater than 5% of the alleles missing for a given SNP, it should be dropped. And that's again an unspoken standard, is greater than 5% missingness. The next is Hardy-Weinberg equilibrium. This is what you learn about in basic genetics classes to see if a given allele is more common than you would expect by chance under a given set of certain conditions that are outlined by Hardy and Weinberg. It's a seminal paper. It's really great. And here we're just actually, we tend to use this test to look for indications of poor genotyping. It's often the case that if a SNP is poorly imputed, a poorly genotyped, the genotype will actually deviate from what you would expect given the distribution of alleles. What I mentioned before, actually, why you need a diagnosis is actually it's really important that you actually do these tests only in healthy controls. It's possible that if you're ascertaining a case control study, for example, and the allele is strongly related to the disease, it'll actually look like the disease allele in the control patients is actually more common than you would expect by chance, and you would think it was a deviation from Hardy-Weinberg equilibrium and drop it. But that's actually one of the SNPs you're actually looking for. So in that case, we actually want to do these tests only in controls. Oh, sorry. Next, and this is really important, is population stratification. The issue of ancestry and comparing different ancestries within a single analysis is potentially problematic because the differences in allele frequencies across patients or subjects with different ethnicities can cause spurious associations to appear. The classic example is from Eric Lander. This is the chopsticks example. So say you want to study a trait, the ability to eat using chopsticks, and you decide to look at the HLA-1A allele in a population in beautiful San Francisco. And you know ahead of time that the HLA-A1 allele is more common among Asians than it is among Caucasians. So when you're looking for an association, when you do a test, you collect samples with from a mixed population of Asian and Caucasian. You would actually come to the conclusion that the Asian ethnicity is associated with the ability to use chopsticks, which clearly is not right. And so that's kind of an easy way of remembering that differences in ancestry can cause you to make conclusions that are not necessarily sensical. So what do we do to avoid this? We use multidimensional scaling. There's a few other tools that are similar, but the basic idea is to compare subjects in your sample to subjects of known ancestry, and then compare the distance to those to assign your subjects a likely ethnicity. And you can do that using multidimensional scaling. I don't think that I added the link on here, but there's a nice set of code on the Enigma website under genetic protocols for running MDS analysis. So I think I apologize for not having it there, but it's all freely available online, the code for and the steps for doing an MDS analysis. After that, I actually have in the at the GitHub address that I showed at the beginning some code for visualizing the results. Yep. So some code for visualizing the results, and actually I think I'll just show you. So each of the points on here that are in red are from the ADNI sample, and each of the others are corresponding to a reference sample of known ethnicity. CEU is corresponding to people of European ancestry, and TSI is a sample from Tuscany in Italy, and our red dots are actually completely covering those two samples here. So that this is about where the CEU samples fall. But you can see there's quite a few red dots that fall within or closer to the reference samples of different ethnicity. So one way to we need to filter them out. We've added in this script on GitHub this sort of sphere that you can use to expand or contract to select only the CEU subjects, and it'll output a set of subject IDs that you can use for the rest of your analysis. One other thing to mention here is okay. So here with the plot name, you split it. put it on plot final equals true, and it'll output all of that, like I said. This is actually the parameter that you will change to expand or contract the circle, so it'll include or be more inclusive or less inclusive. And this is actually, after you do the plot final, you'll see that it now just leaves in the subjects that were inside of that circle before. Right. One other thing is that in this case, in ADNI, the largest single ethnicity was actually people of European ancestry, but obviously that's not the case for all samples. So that's something that you'll need to adjust to your own given data. If you have, say, a large sample of Japanese people, you wouldn't use the CEU sample as a reference. This is just some code for dropping them from your final data set, and you end up with a CEU only, the European ancestry only data set. And now you're finally ready for a genome-wide association study. We showed before actually the toy example of brain size, and that was just at a single SNP. And as you've seen before, or are probably well aware, there's potentially millions of SNPs in the genome. And the mainstay of testing all of these is the sort of exploratory hypothesis of genome-wide association study. This plot on the right is something that you'll probably see a lot if you haven't already. It's called a Manhattan plot. The idea is that each of these sort of look like towers, and the more significant that they get, the more they look like towers. Each point on here is actually the p-value of a SNP. It's a single SNP. And it's plotted as the negative log 10 of the p-value, so the higher up it gets, the more significant it is. Usually when you're testing a single trait, the threshold for significance is 5 times 10 to the negative 8. Again, that's sort of an unspoken threshold. It is based on some theory, but that's the holy grail of getting something significant in a GWAS. So here's some steps for running a genome-wide association study in Plink. It's quite easy, so I don't think I'll spend too much time talking about this, but you essentially just pick an imaging phenotype. It could be any kind of phenotype, really, and put it in a text file and use the --pheno command. And then also you can obviously implement covariates. So you'll create a very similar file to this where each additional column after this would be the covariates of interest, and running it in Plink. I want to show the output, though. So this is actually the association output that you'll get from Plink. And in the example file that we just did, we used sex and age as covariates. So Plink does something sort of strange, although it is useful in its own right. That it gives you a set of p-values for each of the covariates that you've done. So if you do a genome-wide association study, you'll have a p-value for every SNP times the number of covariates that you've included. So it's a huge file. But actually, the line that you're interested in terms of reporting an association is actually the one called ADD, and the p-value associated with it here. These are the p-values of the covariates when the model is fit. But this first line, and then each line after that are the ones that you're interested in. So how do you tell if something's gone wrong in your analysis? This is actually a way of looking at your data to see if they deviate from a null distribution. This can indicate problems like population stratification wasn't properly taken care of, or cryptid relatedness, or problems in ancestry, or just not using the proper model for testing. This is an example of a good plot, but it's actually pretty null. One of the ways that we describe whether or not something is a good plot versus a bad QQ plot is using the lambda inflation factor. A good rule of thumb is to say excuse me, is to say if you have a lambda inflation factor between 0.9 and 1.1, then you're good to go. But in some cases that's not always the case, which I'll show you in the next slide. But here clearly there's a deviation from the null from the very beginning, and that indicates that there's probably a problem in your model, or possibly one of the things that we talked about before. So what about when there's a deviation from the null line because you have really excellent results? That's in that case you'll also have an inflated lambda. One way of getting around this and checking this is actually to exclude your really significant results and then rerun the lambda plot to see the QQ plot to see if your lambda is more close to reality. And that's something that they did in the latest PGC schizophrenia paper, because they did see quite a few significant hits in that paper. The rest of it, and I'm running out of time, and I kind of knew that, so the next few slides are actually describing how you can use your newly cleaned and readied ADNI Plink file to do image-wide analysis. So you can say you're interested, you're following up a hypothesis about a single SNP, and you want to do a point-wise test in an FA map in your subjects. There's code online at the GitHub that I talked about before that will help you go through that process. And the following slides will show you how to do it. And you can end up with pretty pictures like this, for example. Also, I have some good web resources, and I think Jason will actually talk about a lot of these databases for following up something after you've already found it. And then that's thanks for everything. Yeah.
Okay, does anyone have any questions for Derek?
Analysis.
Yeah.
If you do the MDS analysis and using the sphere or the circle to exclude individuals after minor allele frequency, genotype frequency, and Hardy-Weinberg equilibrium, aren't you now changing those three parameters? You're basically dropping, I think in the ADNI1 data, you drop up to about 40 different people.
Yeah. Yeah, absolutely. That's the case. You can again put it through filtering afterwards, and that's probably wise to do. I think especially if you have a smaller sample size, any changes would have a big impact on each of those numbers. If you have a larger sample, I don't think that it would change very much. Another thing too is that to keep in mind is that sure, if you didn't do that, if you didn't do a second round of filtering and you have some results, you should still go back and make sure that each of those things make sense, right? So is the minor allele frequency too low for some reason? Is it, there's still some follow-up that you should do, yeah.
So would it be better to perhaps regress out those first two dimensions from the MDS plot? That way you might be able to keep individuals that are also just not restricted to Caucasian individuals.
That's a really great question, and in general we exclude, and then also we exclude people that are non-CEU for the ADNI case, and then still also include some MDS components. Your question though is whether or not you can control for the different different MDS components and then still leave in the non-CEUs. The problem is that it doesn't always accurately account for the relatedness. It's difficult to account for ethnicity differences using just 4 components is another way of saying that. So there are specific statistical models. This is actually an area of quantitative genetics that's it's a hot research topic, is how you can include data from heterogeneous samples. But definitely the multiple linear regression is not the way to go. In that case, you would want to use something like mixed models and more appropriately accounting for ancestry. Yeah.
Thanks.
Okay, yeah, one more question.
These have been really great talks. I have a question more on the imaging than the genetic side in terms of this processing. So let's say we're following all these best practices where we're pooling data across lots and lots of different sites. And you've shown us how to do the QA across on the genetic side. Well, on the imaging side, even if we take the simplest possible thing like intracranial volume or something like that to measure from a structural image, we still know that there's variations across site by the pulse sequence that you use to acquire that. Even when you're trying to make everything identical, different scanners have slight differences in those metrics. And so they can be also, calibrated and validated across sites, and you can know the differences. But where in this processing do you account for that? Is it at this level where you just put site in as a factor or like acquisition parameters as a factor, or is there something else you can do? There may be a lot of variation that might be really rich to explore also that you want to control that as a nuisance factor.
Yeah, absolutely. It's a really great point. And this is something that we've encountered in the Enigma consortium. So in that case, we're doing meta-analysis. So each of the sites are modeled separately. So in that case, you can, instead of modeling a site in a single model with all the data, you can just combine them separately. And you can use some statistics to come up with a heterogeneity score to look to see how different the effect sizes are across sites. And then if you were to do in a single model some kind of test like that, I would recommend a mixed effects model where you're still fitting the model separately. So you're keeping site as a random effect, which allows you to fit a model separately, and then still come up with an estimate, a single estimate in the model of a p-value and in standard error. But it's a really important point, and I mean that wasn't the focus of this part of the talk, but definitely phenotype harmonization across images is very important and definitely should be accounted for either at this step or by the design of your study, whether it's a meta-analysis or a combined mega-analysis. Yeah.
Just to say, the next talk will be on meta-analysis and imputation, so that will cover some of that too.
Yeah, the reason I brought it up now at this point is that it may be that because the scan manufacturers are getting more and more harmonized, I mean, part of the work of this room and the people who develop these things is helping to push so that the acquisitions are getting more and more harmonized. And it may be that I don't know how to think about this. Do you lose power by keeping it all separate, or could you gain some power if you actually knew exactly more details from the DICOM headers of how things were acquired and could standardize more? Can you do more pooling in a way that's more powerful? I don't even know how to think about that, but it might be helpful.
It's possible, and yeah, like you said, I'm not sure exactly how one would go about that, like to be even more detailed by grouping by slice thickness or something, like like some going down to the actual parameters. I haven't seen any work on that, and I haven't myself experienced anything like that. But it's an interesting point.
Okay, thanks. So thank you, Derek, and we're going to have a bio break for let's say till 10:35, so 15 minutes. So if we could all gather back here at 10:35, and we'll do the next lectures. Thanks, everyone.